Cultures by source and select medically relevant bacteria. So the typical specimens that are received in the clinical lab are vaginal swabs, body fluid culture, respiratory culture, blood culture, stool culture, urine culture, wound culture, and then there's some specialized tests. There's immunological tests, there's molecular ID tests, there's even tests that aren't necessarily related to microbiology, like the fecal blood occult test, but it's still performed in the microbiology area. So we're going to go through some of the typical specimens that we would receive and how we would, in general, start working up those specimens. So starting with vaginal swabs, specimen requirements, swabs should be processed immediately or refrigerated, and you have to use a special like Dacron swab or a swab that's not going to kill the organisms that we're trying to look for. You want to avoid freezing. Typical testing culture is rare. It must be ordered. Gram stain is not usually performed. Typically run a molecular-based vaginal panel looking for the DNA of organisms, of key organisms that we would find in the specimen, or you could perform a vaginal wet mount. Details of both of those are going to be discussed more in molecular diagnostics and advanced microbiology, but I will go through some basics in this course. And then also there's group B screening that's done. So it's a swab that they swab on the outside of the vagina and around the anus to check for that group B. And we would inoculate that swab into a limb broth and then follow that up with a molecular test for the genetic material of group B strep. So the most likely organisms that we're looking for in our typical vaginal cultures would be either Trichomonas, which is a protozoan parasite. So if you look in this picture in the upper right-hand corner, this right here that I'm outlining with the arrow is Trichomonas. It has a flagellum, and if you get a recent swab, you can see these things turning and spinning in culture, so they'll actually be moving. They're relatively large and very obvious in structure, especially if they're motile. Candida albicans that causes vaginal yeast infections. So in a Candida albicans specimen, sometimes you'll see budding yeast. Like here you can see these are epithelial cells. And then around the epithelial cells, you can see this right here is a yeast cell and it's budding. Here's like a baby budding yeast on it. So you can see around here are various yeast cells. And then Gardnerella vaginalis, which is a gram-variable bacterium that causes vaginosis or bacterial vaginosis. And here you can see like a typical epithelial cell, you would have a very even edge, but with a clue cell or with an epithelial cell that's coated with bacteria, the edge of the cell becomes unclear. It becomes really fuzzy because there's so much bacteria on the epithelial cell. It blocks the surface of the cell membrane, so you can't really easily define where the cell membrane is. And all of this is different bacteria that's coated on the surface of those cells. So those are the three main organisms we look for. Trichomonas is the number one highest STD in the United States. So Trichomonas is a sexually transmitted disease, and that's a protozoan parasite. Candida is the yeast, and then Gardnerella vaginalis would be bacterial vaginosis. Respiratory cultures. So sputum would be a deep spit. So you would have to really work up the spit from the lungs as deep as you can get that spit to come up. And endotracheal aspirates are common specimens that are received. They are screened using a gram stain. And we screen them not necessarily just looking for the presence of bacteria, but also to ensure that they're meeting the requirements to work up the culture. It's required that there are less than 10 epithelial cells and greater than 25 neutrophils, also known as PMNs, in order to work up the culture. Why is that the case? Well, if you were to just spit from your mouth into a cup, you're going to have tons and tons of epithelial cells, because your mouth is lined with epithelial cells. All of the internal surfaces, all of the skin that lines the inside of the mouth, that's all epithelial cells. So if we get a spit sample, it's going to have a lot of epithelial cells. which means it's also going to have a lot of normal flora. There's a lot of bacteria and other types of organisms in the oral cavity, especially around the teeth, and that increases if there's any artificial teeth or artificial devices inside the mouth, like braces and such. So, when we look at a sputum sample, we want to ensure that the sputum, that thick, deep spit, is coming from deeper inside the respiratory tract. Because in a sputum culture, we're really looking for those deep respiratory tract infections. So when you do produce a good sputum sample, you're not going to have a lot of epithelial cells. You're also going to have a lot of neutrophils because if it's deep inside the body, neutrophils, that they form pus, they're going to come and start eating up all the bacteria that are present in an active bacterial infection. So if we see a lot of neutrophils, that's indicative of a bacterial infection. So those are kind of the requirements. Respiratory swabs have to be processed within 24 hours if refrigerated, or 2 hours if they're not refrigerated. Typically, we don't order anaerobic cultures. So anaerobic means it's an organism that we're looking for that is killed in the presence of oxygen. We don't usually do, like, we wouldn't do an anaerobic culture on a throat swab or anything like that because the respiratory system contains oxygen. There's a lot of oxygen in the respiratory system, so it doesn't make a lot of sense. The pathogens there would typically not be anaerobes because they would be killed by the environment within the respiratory system. So typical testing, usually pathogens are detected using molecular methods or rapid immunological methods. So culture is pretty rare. A lot of times when we're looking at respiratory samples, we're looking for respiratory viruses, which mostly can be identified using molecular techniques, or we're looking for like a strep throat type infection, and that is diagnosed by lateral flow assay, or LFA. We have those rapid strep tests that have you put the sample in and then you wash it through and it goes past, and there's capture antibodies that will capture the surface antigens if strep is present, causing a color change on the test strip. So that type of lateral flow assay would be performed. Sometimes, though, physicians will order a respiratory culture. And a lot of times, like I mentioned previously, these are going to be sputum cultures, or if a patient has been on a ventilator or an indwelling respiratory device, maybe we'll order something like a swab around that device to check for biofilm formation or to check to see what organisms might be growing in there. Because the longer we have indwelling devices in the body, the more likely we are to develop those types of biofilm infections around those devices. So if a culture is requested, we would plate this on SBA. I do want to mention SBA is a common abbreviation for sheep blood agar, and that's the abbreviation we've been using for the majority of the class. But sometimes clinical labs will call them BAPs for blood agar plate. So you need to know these abbreviations and know them interchangeably. So SBA, BAP, same thing. We're talking about a blood agar plate or sheep blood agar plate. CHOC is short for chocolate. That's chocolate agar. Chocolate agar contains basically boiled up or lysed up red blood cells, so it looks the color of chocolate. It smells rancid. It smells like death. But these plates are going to grow fastidious organisms. Fastidious means those organisms that are really tough to grow or that require extra nutritional requirements. Like back when we talked about Haemophilus species, depending on which one, may require X factor or V factor. When we rupture open the red blood cells and we make this chocolate agar, those factors are readily available in the agar to support the growth of those fastidious organisms. So chocolate is one that we would do. And then MacConkey, if you remember when we talked about MacConkey agar, MacConkey contains crystal violet and bile salts, and that will select against the growth of a lot of gram-positive microorganisms. It also contains lactose, and lactose is used to make it differential. If organisms can utilize lactose, they're going to eat the lactose and produce a pink precipitate on the surface, so they're going to turn pink in color. If they cannot ferment lactose, then they're just going to remain the color that they naturally would be, or they would be like a clear type color on the agar. Excuse me. So, just kind of reminding you about what some of these media are, because you will definitely have to know what the media are and what's in them and what makes them selective and differential, what makes or what they're used for in the clinical lab once you get into like taking, some of the exam questions for this class, but certainly those qualifying exam questions. The sheep blood agar and chocolate agar are incubated in CO2, and McConkey is incubated aerobically. You want to examine plates at 24 and 48 hours. You work up any positives, especially if you see like those alpha hemolytic ones, you want to work those up. No growth at 48 hours, you can throw out the plate. So most likely organisms could be a variety of organisms. If they're beta hemolytic, alpha hemolytic, or a lot of the same consistent organism throughout the plate, then those should be worked up. The source matters. There could be a lot of flora. So that's when we go back to like sputum cultures versus spit cultures. Sometimes certain respiratory swabs will come in and they'll just have a ton of normal flora. So being able to identify normal versus not normal takes some experience and some practice looking at these plates. When you get a blood agar plate, it's very rarely going to be uniform like this. Like there's going to be all sorts of different colors and different stuff on it. But being able to look at the plate, hold it up to the light, see if there's any beta hemolytic ones or if there's anything that looks green and slimy like this, those are ones that are more concerning. Or if we do get like a culture and it comes back like this, where it's like predominantly one thing all over the entire plate, then we could probably assume that that's one that we want to work up and take a closer look at. Blood cultures. So specimen requirements: collect blood into a blood culture tube in a 1:10 ratio, 1 part blood to 10 parts medium. This is one of the most important things that determines the success of the blood culture itself, having the correct amount of blood. So 1:10 ratio is the ratio that you should know for this class and for the qualifying exam. You typically would collect 2 tubes. One of them is an aerobic tube, and the other one is an anaerobic tube. They're collected at the same time and they're processed together. So you collect them from the same vein puncture, you send them down to the lab at the same time. In the lab, we prefer to have 2 sets from 2 different sites on the body. So let's say a phlebotomist draws blood into a blood culture tube and forgets to clean the surface with an alcohol swab. Now we have potential contamination going directly into the blood culture tube along with the blood. And we're collecting 2 bottles from the same site. Now let's say, oh, well, the lab wants another set. So let's just continue to collect from the exact same site. Well, now you're collecting from that same exact contaminated site. But if we have 2 bottles and 2 sets of 2 bottles from 2 different areas in the body, and one of them is positive in one area, and then in a completely different area, it's still positive, we increase the likelihood that yes, this is truly sepsis. Now, with blood cultures, we are testing for sepsis. And if you don't know what sepsis is, it's bacterial growth in the blood. So we have bacteria replicating in the bloodstream. This can be significant and can rapidly lead to the progression to death. There's a lot of bacteria that are called iron chelators. It means basically they gobble up your iron out of your bloodstream. And if they do that, if you know what the purpose of iron is in the blood, it's to transport oxygen. Basically, they're taking away your iron transporting molecules out of your bloodstream. In addition to that, certain types of organisms are going to be shedding endotoxins, which have the potential to cross the blood brain barrier. They can be releasing exotoxins or exoenzymes that can cause broad span damage. And because it's in your central circulation system, those toxins are now rapidly spreading throughout your body at really high rates. The toxins can then damage your kidneys as your kidneys try to deal with them and try to filter them out. And that kidney damage can lead to permanent renal failure. So lots of major complications can happen when a person has sepsis. So we want to make sure that if we get a positive blood culture it's for sure a patient has sepsis, because we don't want to report something and cause mass panic and like an emergency situation if that's not true, if it was just due to contamination. So that's kind of why we want the two sets from two different draw sites. SPS is used as the main anticoagulant because it inactivates neutrophils and complement, which could kill bacteria. So when we draw blood and we're looking for bacteria in the blood, we still have the rest of the blood components there. That includes your immune cells. And if your immune cells are still active, they could kill all the bacteria before, or not all of it, but a lot of it, which would reduce the likelihood that it's going to throw a positive result. So that can cause issues when working those up. So the typical testing, as I mentioned, the tubes, you get an anaerobic and an aerobic one. Typically, we would like a second set from another location on the body as well, but one set will do. The tubes are placed in a blood culture system. This is what one of the blood culture systems looks like, and there's different kinds. And basically, you just pop them in there, and then there's different methods that can detect. One of the common methods is like a gas production. So as the bacteria start to utilize the nutrients in the bottle, they'll produce gas, and that gas will produce a pressure change, and that pressure change can be detected by the equipment. When pressure change happens in the bottle, it's typically because there was bacteria there. So an alarm goes off, you go and figure out which bottle through the alarm, and there's a panel here that'll tell you exactly what slot the bottle was in. You pull the bottle out, and then you work up the blood culture bottle. So if you do that alarm, if it's positive after 5 or more days, usually you'll find Staph epidermidis in there, and that's a contaminant. So that's one of the skin bacteria that grows. It takes a while for it to replicate in high enough quantity to be detected by this analyzer. That's why we say 5+ days. So at that point, you would still work it up, you'd identify it, and then you would report this has been, 5+ days, and it's likely a contaminant. If both tubes or both sets of tubes are positive, it's more likely to be a true infection. If both tubes are positive, the tech should only work up the aerobic bottle, because it's likely that the same organism is going to be in both, and the same organism is going to be why both of them turn positive. So we don't need to duplicate the results when we have both of them being positive. Now, why is that likely? If you got bacteria in the blood, that's pretty rare occurrence. If you get more than one species of bacteria in the blood, that's even more rare. So it's more likely that if you have bacteria in your blood, which is supposed to be a sterile area, it's just going to be one species. So if we have a positive growth on the anaerobic plate and a positive growth on the aerobic plate, we kind of look at both of the plates and say, yeah, the colony morphology is kind of similar. These guys look the same color. They look about the same. You would choose to just work up the aerobic plate. Now, if you do have both plates and they do look completely different, then you may make the decision to go ahead and work up both of them, especially if the gram stains are coming back different or mixed. If it's positive within 5 days, you're going to work it up. Whether there's one tube or both tubes or all tubes positive, you're going to work up any positive tubes according to protocols. So the first step you would do is gram stain, and you're going to immediately gram stain it and record the results in the computer, and that puts it right in the patient's file. That will give the treating physician clues as to what antibiotics they could start on that patient. Now, we're still going to do the follow-up testing. We're going to do susceptibility testing and all that kind of stuff to figure out what the identification is and the exact antibiotics that would actually work to treat. But because sepsis can turn fatal so quickly, we typically want to give them some information right away so that they can get started on some type of antibiotic treatment in hopes to delay the progression of that to death. So, gram stain it and then report what you see. There are molecular tests that are available that can detect organisms directly from a positive blood culture tube. So they have primers that will amplify certain regions of DNA for some of the more common blood isolates. So some of our common blood pathogens can be detected by these systems. Excuse me. If those molecular tests are available, that's great. That'll give us another clue, but we still continue on to work up the culture plates. So then you're going to plate it on SBA, chocolate, McConkey, and ANA. ANA is an anaerobic plate, and then you're going to incubate those in the appropriate environments. Typically, SBA and chocolate's going to be in CO2, McConkey is going to be in aerobic environment, and ANA is going to be in an anaerobic environment. Anaerobic agar, or ANA, it selects for the growth of anaerobic organisms by reducing the oxygen availability in the agar. So it contains specialized substances that will reduce oxygen's ability to be inside of the agar itself. It contains blood to differentiate based on hemolysis. Incubated in anaerobic conditions, and anaerobic is different from incubating with CO2. Some students think that CO2 is an anaerobic environment, so we do need to know the difference with the different types of incubation. So we have your standard aerobic incubator, and this has regular air, typically at 35 degrees Celsius. Then we have a CO2 incubator, and CO2 incubator still has oxygen in it, but it just has some added carbon dioxide because some organisms prefer a little bit extra carbon dioxide in their environment. So oxygen is still present, it's just in a lower percentage. We increase the percentage of CO2, and that creates a capnophilic or microaerophilic environment. Capnophilic is talking about organisms that kind of love carbon dioxide or prefer more carbon dioxide. So that's an environment that has more carbon dioxide. Microaerophilic is talking about organisms that like reduced oxygen and only need oxygen for certain life processes. So in a microaerophilic environment, we're talking about it's an environment that has a reduced level of oxygen. Anaerobic is a whole different type of incubation system. So typically what we would do is we would put it in like a jar or in a case, and that case can be completely sealed off from the environment, and we put in a gas pack before we seal off that case, and the gas pack will exchange the air inside of there so that it eliminates all of the oxygen and changes the environment to an anoxic environment or an oxygen-free environment. So again, very different from incubating with carbon dioxide or in the aerobic environment. And this is just a specialized medium that we use to culture anaerobes. So again, if we have that positive blood culture, we do the gram stain, we're going to plate it on the first day after incubation. So after a 24-hour period of incubation, we're going to check the SBA, chocolate, McConkey, and ANA, and we're going to proceed to identify any positives. Typically, the ANA is going to take 48 hours. We usually wouldn't see a lot of growth right away. Beta hemolytic are typically pathogenic. If nothing grew, you're going to re-incubate. And then day 2, or after 48 hours of growth, you're going to check again and you're going to look for new growth. So on day 1, you're going to report exactly what you see. You're going to type in your little notes into the computer. And then day 2, you're going to come back and type in more notes. And then you'll review your notes and be like, oh, wow, now we got something different growing. If there's no new growth, you're going to save one of the plates and re-incubate it, and then you're going to toss the rest. You're also going to check the ANA plate for growth at that time. And then this is just a picture showing a blood culture in the gram stains and some gram-positive cocci chains here. Most likely organisms could be a variety of organisms. Gram-negative rods are almost always indicative of septic shock. Staph epidermidis is typically a contaminant from the skin. You want to identify all organisms on the MALDI, confirm with gram stain, catalase, oxidase, motility, and other confirmatory tests. We have to have multiple confirmatory tests, even though the MALDI is there. Because if the MALDI comes back with an identification of Staphylococcus aureus and we do a confirmatory test and we see that it's catalase negative, that doesn't match. So we have to go back and kind of rethink things and relook at things. Also, if plates get mislabeled or specimens get moved around in the lab or techs add in the wrong notes to the wrong patient on accident, mistakes can happen. Having multiple confirmatory tests can allow us to track those to prevent us from reporting incorrect information to the physician. Stool culture. So specimen requirements, fresh stool sample transported to the lab within 2 hours if not using a transport medium. Otherwise, if you're using a transport medium, it can be stored for 24 to 48 hours at room temperature. You don't want to freeze stool for a stool culture. You also want to get the stool culture done before antibiotics are started. There's specialized tests for specific antibiotic-resistant organisms, and then there's Clostridioides difficile or C. diff toxins, viruses, or parasites, which can also be tested for in the stool. But we're just going to focus on bacteria that cause infection in the digestive tract. So stool must be plated. So we typically don't do a direct gram stain of stool. Why? Because there's so much stuff inside of stool. There's a lot of normal flora inside of stool. Also, even if you try to do a direct examination for parasites and such, parasites look a lot like partially digested food or whatever type of digested roughage is left in the stool from the sample. So even if we're going to look at stool for parasites, we would have to do a number of like concentration procedures to be able to try to isolate those parasitic forms. So stool, we typically do not look at that directly under the microscope. We're going to plate it to try to see what we have. And when we plate it, we are always going to plate it on SBA and MacConkey. Sometimes physicians will order extended tests, so then we would go ahead and plate it on CVA for Campylobacter, HE, Sorb, and GNBRO. A physician may order additional testing for Vibrio, Plesiomonas, and Aeromonas if infections are associated with exposure to contaminated water. So if the patient has a history of traveling to areas where these things are endemic, if it's suspected that there's like flooding or a contamination of a whole bunch of water somewhere, these organisms are water-loving organisms and found and transmitted through the water. So that would kind of give us clues that we would probably want to use some type of specialized media. TCBS is a medium that's kind of selective for Vibrio. So that's one of the agars that we would use to identify Vibrio in the lab. So if we suspect this type of organism, we would plate it on TCBS as well. So SBA and MacConkey were discussed previously. The SBA, what we would do is when we streak that one for isolation, we would incubate it for a day, and stool typically grows really well after a day. So in 24 hours, you'd probably have a lot of good growth. Then you would perform a sweep oxidase test. So typically in a sweep oxidase, you're going to take a cotton swab and sweep it across the area of the plate that has the most growth. So your quadrant 1, your first quadrant that you did the streak plate for isolation, then you're going to use those drops of oxidase reagent, and you're going to put the drops directly on the surface of the bacteria and look for a deep purple or a dark blue color change. And if you see those color changes, then you know it's oxidase positive, which means it's probably not one of your typical enteric pathogens. It's probably one of the Vibrios, Plesiomonas, Aeromonas. It could be P. ruginosa. So we want to do that to get that initial information. MacConkey, again, I discussed that previously. True pathogens are typically clear, which means the ones that actually cause the majority of the infections are usually not lactose fermenters. CVA, HE, Sorb, and GNBRO, I'm going to talk a little bit more through the next few slides. So CVA for Campylobacter. So CVA contains cefalpirazone, which inhibits most gram-negative organisms, vancomycin, which inhibits gram-positive bacteria, and amphotericin B, which inhibits fungi. And it selects for the growth of Campylobacter. When we streak for isolation on this agar, most other things are going to die, but if Campy was present in the patient sample, Campy would still survive. We also, as another like selective feature, we're going to put this plate in a microaerophilic environment. So we're going to put it in an environment where there's a little bit more CO2 and incubate it at 42 degrees Celsius, so much higher temperature. Hectoenteric agar, or HE agar. This agar is used to kind of specifically look for Salmonella and Shigella. It contains bile salts, bromothymol blue, and acid fusin dyes, which inhibit most gram-positive organisms and non-pathogenic gram-negatives. It also contains lactose, sucrose, and salicin. If they can ferment them, the agar turns yellow. Non-fermenters such as Salmonella and Shigella stay blue or green. So the ones we don't really care about are going to be yellow on that plate. If we see any blue or green ones, those are the ones that we pay a little bit more attention to. It also contains ferric ammonium citrate and sodium thiosulfate, which is used for H2S production. A black precipitate means that it's positive for H2S production. Salmonella would be black. If it's no precipitate, Shigella is indicated. So here's the agar, the HE agar. This is that green or blue color, and because there's no black precipitate, we can safely assume that this is a Shigella type species. If it was yellow, it would be some other type of enteric. Now, E. coli and Shigella are very similar, so this agar is useful in differentiating those two. If you put E. coli or Shigella on the MALDI won't be able to distinguish them. So we typically have to have more tests that help differentiate those. This is one of the agar plates that can differentiate them. There's a bunch of others, but also the Microscan or Vitek 2 analyzer, those two use biochemicals to identify. Those can help differentiate Shigella from E. coli. So structure-wise, they're difficult to identify, but biochemical-wise, we can distinguish them. And then here's that black precipitate for H2S production, which is indicative of a Salmonella species. Sorb agar. So sorb agar is sorbitol MacConkey agar, and this is used to detect enterohemorrhagic E. coli O157H7. This is that really bad strain of E. coli that can eat holes through your intestine and perforate your bowel and go through and cause all sorts of nasty infections in the body. So most strains of E. coli can ferment sorbitol, resulting in pink colonies, but O157 cannot. So you would get colorless colonies on this plate. So the plate typically starts out as pink, but if it's O157, the colonies are going to grow clear, and there's not going to be that pink color on them. So this is indicative of that. O157 isn't the only one that does this. There are a few other species, but typically those can be differentiated through previous tests so that we wouldn't get to this point of putting it on a sorb plate and falsely assuming that it's this. Gram-negative broth, or GNBRO. Gram-negative broth is used because it selects for the growth of pathogens, particularly Shigella and Salmonella. Broth can be used to detect the shigatoxin, which is produced by Shigella, via immunological or molecular methods. And then you subculture it on XLD. So you're going to take a swab of stool and you put it directly in here, and then you incubate it. And then the next day, you're going to pull the swab out and you're going to streak the swab on XLD plate for isolation. And then you can use the broth itself for the immunological or molecular detection of that shigatoxin. So we can either look for the genetic material of the toxin, or we can look for the toxin itself using some type of immunological assay. XLD, xylose, lysine, deoxycholate, XLD agar. Non-Shigella enterics are going to ferment xylose and turn yellow. So the yellow ones we don't care about. Just like with the HE agar back here. Yellow we don't care about. But Shigella on this agar is going to turn red. Salmonella is going to turn black. So why would we use both the HE agar and the XLD? It's just another confirmatory test that we can perform. So sometimes labs will not do the HE and the GNBRO. They'll just do the HE or they'll just do the GNBRO. And then if they do the GNBRO, they will then sub this out on an XLD to get that plate read for Salmonella or Shigella. And then here you can see the plates labeled and what color the different species would be. So the diagnostic progression, you're going to begin with SBA and MacConkey. You're going to incubate that for 24 hours. After 24 hours, you're going to do that sweep oxidase test in the first quadrant of SBA. If it's oxidase positive, further testing to rule out Plesiomonas, Eromonas, and Vibrio is necessary. Pseudomonas aeruginosa is positive as well, so it can impact the results if it's present. If SORB, HE, and GNBRO were ordered, so if we did an extended type of stool culture, SORB, you're going to look for a clear colony. If it's clear, then there's another confirmatory test that you have to perform, and that's called the MUG test. If it's MUG positive, it's likely a case of E. coli 0157. Again, why do we do all these confirmatory tests? To make triple sure that when we are reporting a public health emergency to a physician, it's actually a public health emergency. So as I mentioned with that SORB, other species can also be clear, like I believe Klebsiella is one of them that can be clear on that. And we want to make sure that the clear ones are for sure 0157, so we do this secondary MUG test to confirm that. And I'll go through that procedure in the next slide. XE, or HE, sorry, we're going to look for the green-blue colonies, which could indicate Shigella, or black, which could indicate Salmonella. After that, then you do a rapid urease slant. Urea negative ones are of concern. So if they're urease positive, then we don't really care about them, but if they're urea negative, then we want to work them up further. GNBRO should be subbed out to XLD. XLD red is Shigella, black is Salmonella, yellow is your typical enteric that we don't usually worry about too much. So here's that MUG test. So the MUG test is used to identify E. coli 0157H7. That pathogen produces beta-glucuronidase, which hydrolyzes MUG, and MUG is an abbreviation for a large biomolecule, and that hydrolyzes it into a fluorescent product. And then you use a fluorescent light, so you would put some of the bacteria on the disc, mix it in solution in the bottom here, and then you can see in the presence of a UV light, this would be positive, so it produced that illuminating product, or that fluorescent product. This is positive, this would be E. coli 0157. This one is negative. The urea slant, so can become positive within 4 hours. So if we suspect one of those bad pathogens, we would put it on a urea slant, and this turns pink pretty quickly. So within the same shift, we would typically pull it back out and look at it, and if we see the development of any type of pink color, then we would not worry too much about it. But if it was the urease negative, then those are more likely to be the scary ones. So E. coli, Salmonella, Shigella, Pseudomonas aeruginosa, Klebsiella, Proteus, Helicobacter pylori, typically from gastric biopsies only. Those are the pathogens. The most concerning organisms, true pathogens in stool, are typically Salmonella, Shigella, which as I mentioned is difficult to distinguish from E. coli on the MALDI, Campylobacter, E. coli E0157H7, Vibrio cholerae, and why perform all these tests? We must have multiple confirmatory tests when reporting true pathogens. GI infections have foodborne implications and become a public health emergency. So we really need to ensure that it is what we think it is. Urine culture. going back here. So Helicobacter pylori, I'm going to go ahead and remove that one. I just don't want to give you incorrect information there. Okay, so moving on to urine cultures. Specimen requirements. So most report the urine, or you must report the urine source. So clean catch, catheter, aspirated, because that matters. In urine cultures, we do quantitative testing because different types of urine specimens are going to have different amounts of normal flora, and we have to do it quantitatively to make sure that what we're getting out on the plate is actually an infection and not just normal flora. So you have to report the source of urine. For most of our cultures, you want to report the source. Like stool, you don't report the source because stool comes from the digestive tract. That's pretty obvious where that's coming from. But like wound cultures, you want to report the source, and a lot of other types of cultures, you're going to report the source. Urine is no exception from that. So non-leaking container, if you get a container of urine and it's leaked or it's open, you cannot work that up. You have to reject that specimen. If unpreserved, it needs to be processed within 2 hours. Preserved in boric acid urine tube if extended transport or processing time is expected. So this is what those boric acid urine tubes, they usually have like a gray stopper at the top of it, and those can allow you to test the urine later on. So typical testing, perform a quantitative streak on SBA. The Christmas tree method is one method of doing this. There's also some automated analyzers now that will quantitatively streak a certain amount of bacteria or of samples, on the plate. But the Christmas tree streak is what I learned. So you would put a streak straight down the center, and then you would kind of go back and forth until you streak it for isolation. So a 1 microliter loop is used, and here's where you have to know a little bit of math because the qualifying exam will expect you to know your math and know like how to calculate the quantitative streaking, calculate CFUs, and so forth. So 1 microliter loop, there's 1,000 microliters in 1 milliliter. So if we want to get to CFUs per milliliter, what we would do is we would use that 1 microliter loop, streak it on the plate. We would then count the colonies, and then from there we would multiply the number of colonies that we're counting by 1,000. This will get us to colony forming units, or CFUs per mil. When you have a lot of growth like this, and there's tons and tons, then you can just estimate, and they give you like drop down options where it's like less than 10,000, 10,000 to 50,000, 50,000 to 100,000, 100,000 plus. So this one would probably be just 100,000 plus. You wouldn't have to report the exact number of colonies. But however many colonies you would do, you would multiply it, or you would count, you would multiply it by 1,000. So if you do this streak and only 1 colony grows, then you would have 1,000 CFUs per mil. Then you would streak it onto a urine biplate. So half of the plate is CNA and the other half is MacConkey. This distinguishes the gram-positives from the gram-negative causes of UTIs. So this side is the CNA side. Typically only gram-positives would grow on that side. This side is the MacConkey side, and only gram-negatives would grow on the MacConkey side. Sometimes you can get plates like this where it's growing on both sides, and it could be due to 2 different organisms growing, so there's 2 different things in the urine. Other times you would just have growth on one side or just have growth on the other side. So in clean catch urine, you can expect a lot of normal flora. So clean catch means that the patient just used a cleansing wipe, wiped themselves, and then peed into a cup. Lots of normal flora expected, especially in women. Less than 10 colonies, 10,000 CFUs per mil, you wouldn't work that up. So if you see less than 10 colonies on a clean catch, you wouldn't work that up. It's significant though if you see greater than 100 colonies. And the colonies that you're going to be counting are things that look the same. So you don't want to count like the normal flora background stuff. if you see 100 consistent colonies of the same thing, that's something that you would work up. So 100 colonies means you're talking about 100,000 CFUs per mil, and that's also written 10 to the 5 CFU per mil. So be able to convert to these different notations and be able to do these calculations. So what would this one be at 10,000? 10,000 would be 10 to the 4 CFUs per mil. How do you count that? You count the number of zeros, and that's your exponent. So this one is 10 to the 5 because it has 5 zeros. So that's 10 to the 5 because of the 5 zeros. The 5 indicates the number of zeros. That's your exponent. Same thing here. So that's just kind of an easy way to figure that out. That only works if we're dealing in like tens. If we're dealing with odd numbers, then you have to actually do math. Suprapubic aspirate or catheter urine should essentially be sterile. You should work up any cultures where more than one colony is present and the colonies look consistent. So this is a plate of typical normal flora. So there's a lot of different things. Some of these like squiggly things could be yeast growing because they're really large and they have these spicules sticking out of them. This is typical normal flora. Pathogens you would see are uniform and consistent throughout. And you can see what's growing on this side is different in colony morphology than what's growing on this side. So in this case, we would work up both of these because they are two different organisms and they're growing on two different sides of the plate. Wound and fluid cultures. So specimen requirements, you want to do this prior to any antibiotic treatment. Antibiotics really distort the shape of the cells. So if you're going to do some type of microscopy on them, it's not going to look great. They're not going to grow well on media. So we definitely want to do this before we start antibiotic treatment. You can do swabs, but the problem with swabs, so if you have like, let's say there's a boil on the surface of someone's skin and you want to see if there's bacteria in there. What type of bacteria is growing inside of that boil, you would have to take the swab and get it into and underneath the skin to get to the pus region of the boil. If you just try to scrape the top surface of the skin, you're going to get a lot of contamination with Staphylococcus epidermidis, and that's your normal skin bacteria, and that's going to falsify the results. So swabs kind of adhere to the same requirements as when I was talking about sputum. You want to see less than 10 epithelial cells, greater than 25 PMNs in order to work up any type of swab. So it's not really preferred when we're talking about wound cultures. The preferred specimen would be a syringe of pus because now you've aspirated it using a needle. The likelihood that you're going to have epithelial cells is pretty low, and you're going to have purified bacteria and neutrophils present. Tissues can be used, so you can get a chunk of tissue and culture it. When you do that, typically you're going to put the tissue in a little bit of saline, and then you're going to use a tissue grinder. And the tissue grinder, you just manually grind up, and it's this apparatus that you just twist. And once it's all ground up, then you're going to use a pipette to take the solution or the liquid part of the ground up tissue, and you're going to put a drop of that solution on the microscope slide, and then you're going to put a drop of the solution on each one of the plates that you're going to be inoculating. And when you do that, then you go and streak it for isolation. Following the streaking for isolation, you're going to then work up the plates that you see and do your gram stain and look at it under the microscope. Fluids. For fluids, source definitely matters. So like I mentioned before, we want to report source on a lot of different things, but fluids, the source matters. If it's a sterile environment in the body, then we would treat it as a sterile environment, which means we would expect that there's no normal flora growing in that area. So anything that is growing would be assumed to be a pathogen. CSF is a critical. So there's very few like critical or stat things in microbiology, but cerebrospinal fluid, CSF, and blood, those are two very critical areas of the microbiology lab. So for CSF, we would do a cytospin procedure because CSF can be dilute and there could be very few bacteria found, in a wide area of cerebrospinal fluid. So if we get the fluid, we want to condense it down to the bacterial essence that's found inside of the fluid. So this is what that looks like. We would put a microscope slide on here, and we would put the tube with the specimen that we can then spin down, and it would spin it and concentrate it onto one tiny little spot on a microscope slide. And it's like a special centrifuge thing that spins it around really fast. And that way all of the solid components inside of the CSF would be concentrated down to a tiny little spot on the microscope slide. And that makes it very easy for us to be able to identify any type of bacteria inside of that spot. With cerebrospinal fluid, because it is so critical, if we could only get like a tiny bit, like if there's just 2 little drops of cerebrospinal fluid, the first drop, you would definitely do a gram stain. The second drop, you would want to inoculate it onto chocolate agar, because chocolate agar is going to grow basically everything. So if you only have like a very little amount, grow it on chocolate agar. Once you get something to grow, then you can kind of work it up from there. Cerebrospinal fluid, just like blood, is supposed to be sterile. So if there's anything growing in there, it's usually just one thing, and that one thing is causing the issue. If you have more than one thing growing inside of cerebrospinal fluid, that's a very unlikely and very concerning type situation. So sterile body fluid, you want a gram stain, and all organisms are significant when it's coming from sterile body fluid. So the typical testing, you do a gram stain. You need to report white blood cells and epithelial cells per view. So here you can see white blood cells are going to be these bigger pink ones. In the background, you can see some bacteria here. So that looks like gram-negative diplococci. And then these bigger ones are going to be your epithelial cells. So you definitely want to report what you're seeing. 0 to 1 would be considered rare, 1 to 4, few, 5 to 10, moderate, 10 plus, many. And usually when you're putting the results into the computer, there's a drop-down menu and it will give you these options. You're going to streak the fluid onto SBA, chocolate, CNA, and MacConkey. Streak onto ANA if anaerobic culture is requested. Chrome agar can be used for some antibiotic-resistant strains, such as MRSA. So chrome agar is a primary isolation agar, and it produces predictable colors that help distinguish organisms. This is what MRSA looks like on chrome agar, or on this type of chrome agar. But depending on what type of chrome agar you have, different organisms are going to appear different colors. So reporting the results, it could be a variety of different organisms. Anaerobes and beta hemolytic species are the most concerning. There are specialized tests for antibiotic-resistant bacteria such as MRSA. In addition to those chrome agar plates, there's molecular tests that can be performed. And then you want to quantify the growth. So 1 plus all the way up to 4 plus, depending on which quadrants it grew in. So if it only grew in the first quadrant, you'd give it a 1 plus. If it grew in all 4 quadrants, you'd give it a 4 plus. So that just gives you a basic overview of some of the tests that are performed in the clinical laboratory. You will actually be, or clinical microbiology laboratory, you're actually going to be able to perform some of these once you get into your clinical rotations.